Welcome back, my students. Looks like we're really gaining momentum here. We're now in chapter 23 and now part B. That's the digestive system, and a few more. We barely have a month, more than a month. We barely have a month and a half more to go, and the semester is over. So let's take a look at now the pharynx and the esophagus. And pretty much, guys, the pharynx is that funnel shaped organ that lies at the back of our mouth. Well, actually, when food passes from the mouth to the oropharynx, it gets into the laryngopharynx. So it has actually three parts. So the pharynx allows passage of food, fluids, and air. And pretty much it's made up of stratified squamous epithelium, and it has mucus producing glands, lots of goblet cells there. And the external muscle layer consists of two skeletal muscle layers. Okay, you have an inner layer of muscles that runs longitudinally, and then an outer pharyngeal constrictor that encircles the wall of the pharynx. This is the one that helps pretty much in our swallowing. So the esophagus now, next to the pharynx, distal to the pharynx, you have the esophagus, which is a flat muscular tube that runs from the laryngopharynx to the stomach. And the esophagus is pretty much collapsed when it's not involved in food propulsion. So it pierces right through the diaphragm at the esophageal hiatus. And it joins the stomach at the cardial orifice of the stomach, and then pretty much there is a sphincter, or which you call the gastroesophageal sphincter or the cardiac sphincter, that surrounds the cardial orifice. It keeps this hole or orifice closed when food is not being swallowed. So mucus cells on both sides of the sphincter would pretty much help protect the esophagus from any acid reflux. So the esophagus has four alimentary canal layers. It will have the mucosa, which contains stratified squamous epithelium, and then it gradually changes into simple columnar epithelium the moment it reaches the stomach. The esophageal glands in the submucosa of the esophagus secretes mucus to aid in the movement of the bolus. And then the muscularis externa, which is the skeletal muscle, is it's actually like a skeletal muscle on the upper area of the esophagus, and then it's a little bit mixed in the middle, and then it's totally smooth muscle inferiorly. So there is a gradual change from muscular to, so that it'll help you aid in your voluntary swallowing. And then, since you're not swallowing anymore all the way down to the esophagus when pushing the food distally, it's already the peristalsis of the esophagus that does that, which is involuntary. It would have adventitia instead of a serosa. It doesn't have a serosa as a cover. So this is now a cross section of the esophagus, and you can see here that the mucosa. The mucosa of the esophagus, well, you will have to look at it this is actually a scanning view. You will have to look at it under high power magnification to see rather that it's actually made up of stratified squamous epithelium. And here you go, you could see it a little bit closer. It's made up of stratified squamous epithelium right in the area where you have the esophagus. But because this is right at the junction of the esophagus and the stomach, right here, if you're going to take a look at this under high power magnification, you'll find out that these are all made up of simple columnar epithelium. So let's take a look at some homeostatic imbalances. Heartburn. A lot of people suffer from chronic heartburn. It is caused by the stomach acid regurgitating into the esophagus, which is pretty much a symptom of gastroesophageal reflux disease, which is GERD, which is pretty much like what your professor has. I do have that because I have what you call hiatal hernia, which means the gastroesophageal sphincter of my gastroesophageal sphincter is not anymore closing properly. Okay, and of course, when I eat a lot, okay, if there's going to be excessive food, and of course the type of food that I eat, drink, okay, could also pretty much like affect my heartburn. Also, it can be caused by, well, that's it. That's what exactly what I have, the hiatal hernia. It's an abnormality where part of the stomach actually protrudes above the diaphragm. Actually, in my case, really, my sphincter is not really properly closing anymore. That's why the stomach protrudes above the diaphragm. And so I constantly have esophagitis, esophageal ulcer, but I know that's pretty much like um, pretty much like helped by an antacid. So let's take a look at the digestive processes of the mouth. The pharynx and the esophagus pretty much are passageway. They are conduits to pass food from the mouth to the stomach. Major function of these organs will be just to propel, and it starts with swallowing, okay? And it involves coordination of about 22 muscles and two phases. And the two phases will be your buccal or the buccal phase, which is voluntary contraction of the tongue, and then the pharyngeal esophageal phase, which is the involuntary phase that primarily involves the vagus nerve. It is actually controlled by the swallowing center in the medulla and the lower pons. And here you could see the steps in the deglutition or swallowing. You have the buccal or the buccal phase, where you have the upper esophageal sphincter is actually contracted. And okay, let me just get my okay, buccal phase is contracted, and there you go. The buccal phase is actually contracted. The upper esophageal sphincter is contracted and it's actually closed. Now the tongue presses down again very hard against the palate, forcing the food bowels into the oropharynx. And in this section right here, okay, you call this the pharyngeal esophageal phase. The tongue blocks the mouth. You can see that the tongue is a little bit raised here. The soft palate and its uvula would rise, closing off the nasopharynx. The larynx rises also so that the epiglottis blocks the trachea, and you could see it right through here. Okay, without that, without reflex coming down, then you are going to be, uh, you might be like getting food into your windpipe, and that thing is not going to be good. So here the larynx rises also so that the epiglottis can block the trachea, and then the upper esophageal sphincter would relax and the food enters the esophagus. And finally, when the food is right there at the back, which is already the esophagus, you could see that the pharyngeal esophageal phase, the constrictor muscle of the pharynx will contract, forcing the food into the esophagus inferiorly. The upper esophageal sphincter contracts after the food enters the esophagus. And right in this side here, on this lower thing here, is the food is now in the esophagus. The diagram shows us that peristalsis is actually the pinching effect, the circular and longitudinal muscles working coordinately to push down the food down through the esophagus and then into the stomach. Here, the gastroesophageal sphincter opens, so, and pretty much it opens the cardiac orifice, allowing the food to get into the stomach. All right, I went back a little bit there, and that's way too much. All right, and this is pretty much what we have discussed already. So we get to the anatomy of the stomach, gross anatomy of the stomach. The stomach is just a temporary storage. It's a tank that starts pretty much, it's not really a useless tank. It's not just a holding area. It is already starting chemical breakdown of protein digestion. It converts the bolus of the food to a paste-like kind. So here's actually the deal in the way we call or we pretty much like give names to the food that is actually going through our gastrointestinal tract. If the food gets into our mouth, you still call it food, but the moment it mixes with the saliva, you call it bolus. The moment the bolus lands in the stomach and gets to be mixed with the gastric juice, you don't call it anymore gastric juice, you call it chyme. Because right there in the stomach, the bolus will be converted with the help of the um, with the help of the uh, chemicals in the stomach into a paste-like chyme. Okay, an empty stomach usually will have about 50 ml volume, but can expand to about 4 liters. You can just imagine how much food really you can take in. 4 liters. And no wonder there's actually like winners of those Coney Island hot dog eating contests by Nathan's, right? Because they can really expand their stomach up to 4 liters. So when empty, the stomach mucosa forms many folds, which you call ruga or rugae, and the major regions of the stomach will be your cardiac end, which is the one that would be the most superior, which is pretty much like immediately uh, almost like under the diaphragm, and that surrounds this part is the one that surrounds the cardiac orifice. The fundus is actually that particular dome-shaped region beneath the diaphragm. It looks like a pregnant belly, and then the body is the middle portion of your stomach. The pyloric part is the wider and more superior portion of the pyloric region. The antrum narrows down into the pyloric canal and terminates in the pylorus. The pylorus is continuous with the duodenum through the pyloric valve, which is a sphincter that controls the emptying of the stomach. And here you are. This is just very, what do you call this? Very big for me to be transferring to the other slide. But pretty much it tells you where the cardiac end is. It is the cardiac end right there. Then the fundus will be this enlarged area here. Then the body is here. Then you have the pylorium, the antrum first, and then the pyloric sphincter is right around this area here. And the stomach, is curved. It's a J-shaped organ. You have the lesser. This is what you call the lesser curvature of the stomach. And you have this part here to be the greater curvature of the stomach. You will find out, or we will learn later on, that the lesser curvature is actually connected to an omentum that will hold the stomach to the liver. You call that the lesser omentum. And this greater curvature will pretty much like be connected to another wider omentum that would connect the stomach, the greater curvature of the stomach, to the transverse colon. And this particular omentum is named the greater omentum of the stomach. And here's actually our picture of the stomach with your telling us exactly where the greater curvature is. And I've already discussed it. And the lesser curvature, that's the lesser curvature and the greater curvature. All right. And the mesenteries will be your greater omentum and your lesser omentum, which is actually very visible in there. Right here, this area right here, beautiful. It's an omentum that connects the lesser curvature of the stomach to the inferior surface of the liver. Okay? And right here, and the greater curvature, you could see this fatty peritoneal extension, and this is actually your greater omentum. So the greater omentum drapes inferiorly right there, okay, connecting the greater curvature to the transverse colon. Pretty much this blends with the mesocolon, mesentery. It's a mesentery that anchors the large intestine to the abdominal wall, and it contains very plenty of fat deposits and lymph nodes. As a matter of fact, uh, this would typically become part of the beer belly whenever we eat so much beer or drink so much beer and eat a lot of food. So, what is the nervous supply of the stomach? Well, sympathetic fiber from the thoracic splanchnic nerves are actually relayed through the cardiac plexus. The parasympathetic fibers are supplied by the vagus nerve. And the blood supply pretty much is coming from the celiac trunk, okay, and from the gastric splenic branches and the veins of the hepatic portal system. Okay, now we go a little bit into the deeper, which is actually on the microscopic part. The stomach wall has four layers or four tunics, and pretty much you will have the mucosa, okay, the mucosa, the submucosa, the muscularis, and then you have the serosa. Okay, your stomach has a serosa. It does not have an adventitia because it is actually inside the abdominal cavity and is actually separated. So it should have its own peritoneal fold, a visceral peritoneum. So the muscularis externa, which is the muscle layer, this is the entire muscularis uh, of the muscularis layer of your stomach, would have three layers, contrary to the two layers that are found mostly in the tubes of the gastrointestinal tract, such as your esophagus, your intestines, and the colon. These tubes have only two muscularis externa. It will have an outer longitudinal, they all have outer longitudinal and another layer of inner circular layer. This stomach has an extra one, which is the oblique layer. So the presence of the oblique layer will allow the stomach to churn, to mix, to move the chyme, to move three-dimensionally, so that it can also pretty much like pummel the food that is inside, which pretty much would increase the physical breakdown and and then ram it down into the small intestine so that it can be moved into the small intestine. The mucosa, which is the one that covers the lumen, is also modified. The mucosa contains simple columnar epithelium, which is a drastic change from the stratified squamous epithelium of the esophagus. Okay, and they are made up of many goblet cells or mucus cells. They secrete two-layer coats of alkaline mucus. Alkaline mucus would help, the coat of alkaline mucus would help preserve the stomach lining and pretty much like put it into uh, safety whenever the stomach is actively producing gastric juice. So the surface layer traps bicarbonate-rich fluid layer that pretty much is beneath it. It is also dotted with gastric peats, which lead into gastric glands. You could see that each of those are the pits, but when you go right through where the pits are going straight down, it goes straight down. Okay, the neck is filled with uh, mucus cells of the neck. Then it goes down into having more of the gastric chief cells and the parietal cells. So the gland cells, let me just be more specific with it. This is actually now just a version, another version of the picture that I had in the previous one. Here you could see the mucous neck cells, okay? You could see the parietal cells. These are the cells that produce hydrochloric acid, okay? And what do you call this, the intrinsic factor. And then these are also the chief cells, and then you have the enteroendocrine cells right there, okay? So pretty much these are all glandular and secretory in nature, okay? And each of them has their own specific function, and we're going to get into them. So the mucous neck cells, these are your mucous neck cells right there. Those pink ones are the mucous neck cells. They secrete very thin acidic mucus, which is pretty much like of a known origin, okay? The parietal cells on the other hand, the blue cells right there, okay, are the ones that would be secreting hydrochloric acid, and therefore it creates a very acidic pH in the stomach from 1.5 to 3.5. It helps denature protein. It activates pepsin. See, pepsin gets to be pepsinogen, which is a precursor, gets to be activated into pepsin under a pH only, an acidic pH of between 1.5 to 3.5. Without that acidic pH, pepsin is not going to be formed in the stomach. Okay? What's the purpose of pepsin? Pepsin is supposed to be a protease. It is a protease, and it serves to um, initialize or commence or start the breakdown, the chemical breakdown of protein. So for people who are constantly creating an environment of not being so acidic in the stomach, because they want to take lots of antacid because of heartburn, then they are not going to be like effectively um, uh, chemically digesting their protein in the stomach. But nevertheless, when the undigested protein gets into the small intestine, then the small intestine will continue on and will be completing the digestion, hopefully. Okay, that's why people who have heartburns are pretty much like, um, or would need to pretty much like control the way they're, or the amount of protein that they're, and fat that that's actually in their diet in order to prevent the production of too much acid in the stomach. Then you have your intrinsic factor, which is actually a glycoprotein that is necessary for the absorption of vitamin D. Okay. So chief cells. The chief cells is one of the major secretory cells, and they pretty much like secrete pepsinogen. Pepsinogen is the inactive form of pepsin. It has to be activated into pepsin by hydrochloric acid. And the chief cells also will be producing lipase, which is uh, which can actually digest fat, but because there is no emulsification of fat in the stomach, then only about roughly 15% or less of the lipids is actually being digested. Only the small triglycerides are being digested by the lipases in the gastric juice. So, the enteroendocrine cells, all the way at the bottom, you see all the green ones. These are uh, uh, pretty much cells that would secrete chemical messengers or hormones into the lamina propria. And these hormones act as paracrines. They secrete serotonin and histamine. And pretty much they also secrete hormones. And the hormones are somatostatin, also known as paracrine, and gastrin. So, this is a closer view, pretty much, of one gastric pit. It's a bigger version. You could see your parietal cells, and these parietal cells secretes HCl and the intrinsic factor. The chief cells would secrete pepsinogen, which needs to be activated into pepsin by the HCl of the parietal cells. And then the enteroendocrine cells secretes hormones and paracrines. The hormones and the paracrines are actually also on a feedback loop, will be stimulating the what you call is the production of HCl and the production of pepsinogen as well. So your stomach has your mucosal barrier, pretty much, and the purpose of this is that is actually to protect the stomach whenever harsh digestive conditions pretty much would require the protection of the stomach. So this mucosal barrier protects it, and is created by three factors. The Pretty much you have one of the three factors is a thick layer of bicarbonate-rich mucus, the tight junctions between the epithelial cells, pretty much which prevent the juice from seeping underneath the tissues, And pretty much damaged epithelial cells are quickly replaced by the division of stem cells. And really, they are really very quick in replacing damaged cells pretty much like almost on a daily basis or each time, every time we eat. There are lots of epithelial cells that are damaged and therefore they need to be replaced as fast as they are torn out. So they are now going to be completely replaced sometime like every 3 to 4 days or 3 to 6 days. And here's a picture of an ulcer. Before we get into the ulcer, let's take a look at one homeostatic imbalance, which you call gastritis. It's an inflammation caused by anything that breaches the stomach mucosal barrier. Okay, and here I got lots of arrow right there. This is a picture of gastric ulcer. This can be caused by an erosion in the stomach wall, and if the erosion perforates the wall, it can lead to peritonitis and hemorrhage. So most ulcers are caused by a bacteria, which you call H. pylori, Helicobacter pylori. And it can also cause by non-steroidal anti-inflammatory drugs, such as aspirin. And this is an electron micrograph of the bacteria, the H. pylori, okay, that is actually found in the stomach of people that has gastric ulcers. So let's take a look at the digestive processes in the stomach. What are the things that are happening in the stomach that aids in the digestion, or that's particularly or pertaining to digestion itself? Here are the processes where the stomach carries out the physical breakdown of food. Well, of course, you break it down with your tongue, between your tongue and your teeth, but it is going to be further broken down in the stomach. And it also serves as a holding area for food. It delivers chyme to the intestine, partially digestive, chemically digestive chyme into the small intestine. It denatures protein, okay, by secreting hydrochloric acid. And then pepsine, once the pepsinogen is converted to pepsine, pepsine carries out enzymatic digestion of proteins. Now there's also the milk protein casein, which is broken down by renin also in infants, and renin is produced in the stomach by infants. And that's the reason why it results in curdy substance. So when infants really would accidentally throw up, then they throw up really like a curdy substance. It's because of the renin secreted by the stomach, which is partially curdling the milk that they have taken in. Okay, so there are some drugs that are absorbed in the stomach. For instance, lipid soluble alcohol and aspirin are absorbed into the blood through the stomach. And only stomach function that's essential to life really is the secretion of intrinsic factor for vitamin B12 absorption. Whenever you are unable really to produce or intrinsic factor for vitamin B12 absorption, you're not going to be able to absorb this. And so you're going to have some problems with your red blood cell production. You're going to be anemic. And so in such cases, because your B12 is needed for your red blood cell maturity, it can cause pernicious anemia. And usually it could be treated very well with B12 injections. Okay, let's take a look at the gastric secretion. The gastric mucosa secretes about roughly more than 3 liters of gastric juice per day, and they are regulated by neural mechanisms. You know that when we are in a parasympathetic mode, that's the time we can pretty much like digest our food. And the main branch of the parasympathetic nervous system, and we know this from our what we call this nervous system in AMP1, okay, is actually the vagus nerve. The vagus nerve runs all the way. It's actually runs all the way from what do you call this, from the cranial nerves. It's actually cranial nerve number 10 all the way down and um, would be innervating almost about 85% of the gastrointestinal uh, tract. I mean, it goes down. So sympathetic stimulation, the vagus nerve is actually the one that promotes digestion. The sympathetic stimulation will be doing the opposite. This is the one that secretes, that decreases the secretion, and therefore it does not promote. It inhibits digestion. Now, in the hormonal mechanism, gastrin, a hormone gastrin, is the one that stimulates the hydrochloric acid secretion by the stomach. And gastrin is an antagonistic hormone, okay, secreted by the small intestine. They are, pretty much like antagonistic in action. So gastric secretions are broken down into three phases. First of all, in the cephalic phase, in the gastric phase, and in the intestinal phase. These are the three phases of gastric secretions. We're now going to discuss what these three phases are. In the cephalic phase, this is the phase where conditioned reflex is triggered by just the mere smell of food, or the sight of food, or even the thought of food, or even just a taste it. Remember, you are not swallowing anything here. Okay, so the sensation pretty much would stimulate the medulla oblongata, right there, the medulla oblongata. Okay, and then it's going to be delivered down through the vagus nerve by the parasympathetic action, and pretty much those postganglionic neuron will stimulate the secretion by the parietal and the chief cells, and stimulates the production of gastrin and histamine by the endocrine cells. Gastrin is carried through the circulation back into the stomach, okay, where along with histamine, it's actually going to, there you go, along with histamine, it's going to stimulate secretion. That's really a lot of things happening even without food getting into your stomach. That's a lot. So pretty much, um, in the gastric phase, okay, in the gastric phase, which is the second phase, this time you already have food. You already swallowed food. You ate food. Then pretty much you put food, and so food landed on your stomach. So here the distension of the stomach, okay, right here. Okay, here the gastric phase is actually the phase that lasts 3 to 4 hours, and pretty much provides 2/3 of gastric juice that is released. And the stimulation of the gastric phase would include distension that activates the stretch receptors, initiating both the long and the short reflexes. So right here, okay, in this particular model right here we have in front, the distension of the stomach, okay, so here is actually your distension, the blue arrows right there, would stimulate the receptors of the stomach and would activate the parasympathetic reflex. And pretty much, as it activates the parasympathetic reflex, action potentials that are generated by mechanoreceptors are carried by the vagus nerve into the medulla oblongata. Okay, and that's exactly how it goes right through. The medulla oblongata increases action potentials in the vagus nerve and stimulates secretion by the parietal cells, okay, and the chief cells. And it will stimulate also the release of gastrin and histamine by the enteroendocrine cells, like this one here. Now, the distension of the stomach also would activate local reflexes, pretty much, that increases stomach secretions. And that's actually going in that particular direction. Gastrin is carried through circulation back to the stomach, okay, and that's exactly going around, okay, where along with histamine it would stimulate secretion. So, okay, How is the gastric phase really stimulated? Well, the release of gastrin initiates, well, I mentioned that to you already, initiates the HCl release from the parietal cells and activates enzyme secretion. Okay. It prods the parietal cells to release HCl by binding to receptors on the parietal cells and stimulating the enteroendocrine cells, right, in this section to release histamine. Okay. There's also the buffering action of proteins that we ingest, causing the pH to rise, which activates more gastrin secretions. So the gastric phase pretty much fills our stomach. It fills our stomach up to a point, right, in this section right here, where when the stomach reaches this point, there's going to be a point where the pyloric sphincter will now open. When the pyloric sphincter opens, it will slowly empty a little bit of chyme into the duodenum. The moment there is actually a little bit of chyme that gets into the duodenum, then there's going to be an inhibitory loop that's going to be sent into the stomach, signaling what you call is the start of intestinal digestion, and pretty much like the possible slowdown and end of the stomach digestion. So whenever the first chyme reaches the duodenum, that's when we pretty much practically feel we are already full. So the faster you're eating, okay, it actually gets into that particular part of the pyloric end, right, almost like at the same time from the start of eating food, about 10 to 15 minutes or 20 minutes, it goes right there. So if you eat fast and you fill your stomach faster than 20 minutes of eating, definitely before that first drop of chyme gets in there, your stomach is already full, which means in order for us to really lose weight, we have to make sure that we eat slower. If we eat slower, it will give us time not to be filling the stomach, but enough time for the chyme to get in there. And then once the chyme gets into the duodenum, that will slow down our stomach and will make us feel full. And if we eat slow, then we are not going to be filling our stomach so much, and yet we already feel full. So if we eat fast, our tendency will be to load our stomach fast, loader than the chyme, the length of time that the chyme can actually uh, begin to help inhibit the action of the stomach. So we have to slow down in our manner by which we eat in order that we are not going to be feeling ourselves too much. So right here, low pH would inhibit uh, gastrin secretion. It occurs between meals and occurs during digestion as a negative feedback mechanism. The more protein and the more HCl here is actually secreted, causing a decline in pH, which inhibits gastrin secretion. These are one of the things that would inhibit gastric secretion, and it occurs during or in between meals, okay, or even during digestion as a negative feedback. If you eat more protein, okay, and you have a lot of HCl that is being secreted, it causes the acidic pH would pretty much like inhibit gastrin secretion. Now we get into the intestinal phase, and this is the phase where the first drop of chyme in the duodenum, or the first drop of chyme reaches the duodenum. So chyme, and you call that the intestinal phase. It begins with a brief stimulatory component, okay, which is brief stimulation followed by inhibition. That inhibition pretty much will slow down completely the stomach, and that will give us the feeling of satiety or being full already. Okay, so pretty much the stimulation of your intestinal phase is because of the partially digested food that enters the small intestine, which you call chyme, causing a brief release of intestinal gastrin. That's actually the stimulation phase, but that is only very brief. It's going to be followed by um, um, a component of inhibition. So, in the inhibition process, inhibition of the intestinal phase, there are actually factors in the duodenum that causes inhibition of gastric secretion. First of all, you have the distension of the duodenum, okay, due to the entry of chyme. So pretty much your duodenum will be receiving a lot of food and gets to be distended. And then the presence of the acidic chyme, okay, and the presence of fatty acid chyme also, and the presence of hypertonic chyme. All of these are actually factors that will cause the inhibition of gastric secretion, which means, you know, that you have already reached that point where there's so many food already in the small intestine, and the stomach digestion needs to slow down. And when the stomach digestion has slowed down, that's already a cue for us in our brain to feel that we are already full. Okay, the inhibitory effects protect the intestine from being overwhelmed by too much chyme or too much acidity from the chyme. Because if there's going to be a bulk of food that's going to be in the stomach that needs to be emptied into the intestine, it has to be emptied very slowly in really very small spurts only. The spurts are actually like, if I'm not mistaken, it's only like three contractions of the pyloric sphincter per minute. And that's going to be like uh, slowly um, releasing the acidic chyme into the duodenum. That will give the duodenal cells, the duodenal glands, the chance to secrete lots of mucus that is rich in bicarbonate. So the Brunner's gland or the duodenal glands are the ones that are aiding in the in neutralizing the acidity of the chyme. But that's only one. That's only one help. The big chunk of of neutralizing the acidity of the chyme is actually the bicarbonate rich secretion coming from the pancreas. So right here, before we get into that intestinal part or the intestinal digestion, take note that there are actually like chemoreceptors in the duodenum, okay, that are stimulated because of the acidic chyme that enters it. And action potentials generated by the chemoreceptors are carried by the vagus nerves to the medulla oblongata, right there, where they inhibit parasympathetic action potentials, there you go, like in a feedback loop, by decreasing gastric secretions. There are also local reflexes that is affected by the presence of hydrogen ion or lipids, which also inhibits gastric secretion. And pretty much, this is actually the loop right there, the orange loop right there. Okay, and then you have secretin and cholecystokinin. These are hormones that are produced by the duodenum. Okay, and they decrease gastric secretions in the stomach. Okay, so that's your brown loop right there, secretin and cholecystokinin. And that particular secretin and cholecystokinin action is what you call the what you call is the enterogastrones. These are coming from the duodenum, and they are released by the enteroendocrine cells. They inhibits gastric secretion. So here's actually one of the cheat table that you can work around with. Take note. This is a really very important for your review. These are the inhibitory events, and they are the pattern of the arrow will be broken arrows for inhibitory events, and solid arrows for stimulatory events. Okay, so this is just a repetition of what we have done. So you could use this as part of your review. And just is a really list down table of intestinal juice and gastric juices and their actions. So we're going to just a little bit, okay, we're not going to go so deep into the mechanism of hydrochloric acid formation, but we just want you to know, to add up, right, that the parietal cells are the ones that are responsible for producing HCl. The parietal cell pump hydrogen ion from the carbonic acid breakdown, okay, into the stomach lumen via your hydrogen ion potassium ATPase proton pumps. And as the hydrogen is pumped into the stomach lumen, the bicarbonate is exported back into the blood via the chloride and bicarbonate antiporters. So there's actually an exchange. Now this results in the increase of bicarbonate in the blood. Remember that the bicarbonate is negatively charged, leaving the stomach, okay, and this is actually referred to as, because it leaves, it forms what you call a tide. And because your bicarbonate is alkaline, then it creates what you call an alkaline tide. There is also a chloride that is pumped out to the lumen to join the hydrogen ion, and thus forming hydrochloric acid. You're probably wondering, hydrochloric acid is an inorganic acid that is really very, very acidic. It's a very strong acid. How the heck are we forming this? Well, this is exactly how we form our hydrochloric acid. And pretty much this is exactly how it is. All right. First, you have your hydrogen and your bicarbonate ions that is actually generated from the dissociation of the carbonic acid. Okay. There you go. From the dissociation of the carbonic acid right there. All right. Then you have your ATPase pumps into the lumen. Okay. And there you go. They're being pumped. All right, and finally, the bicarbonate leaves the cell, as in the alkaline tide. Okay, this is your alkaline tide as it leaves the cell and gets into the bloodstream. That's your alkaline tide. And then finally, your chloride diffuses into the membrane. There you go. The chloride diffuses into the membrane, gets into the, okay, there you go. There, gets into the membrane, and here HCl is produced. So how is it regulated? What's regulating the gastric motility and emptying? Well, as a response to stomach filling, okay, the stomach stretches out to accommodate the incoming food. And two factors will cause the pressure to remain constant until you have about 1.5 liters of food that you have ingested. There is actually receptive relaxation, which is reflex-mediated relaxation of your smooth muscle, and is coordinated by the swallowing center of the brain. The ability of the stomach to relax when filling with food is actually this receptive relaxation. Then you have gastric accommodation. This is the intrinsic ability of the smooth muscle of the stomach to pretty much like exhibit the stress relaxation response, which enables the hollow organs to stretch without increasing tension. So we are actually allowing our stomach, in short, this receptive relaxation and gastric accommodation allows our stomach to stretch and accommodate food, okay, and pretty much up to about 1.5 liter of food is actually taken in. So how is motility and emptying regulated? Well, peristaltic waves move toward the pylorus at the rate of about 3 per minute. Okay? And there's actually what you call the basic electrical rhythm, or the burr, is set by enteric pacemaker. Okay? And these are linked by gap junctions to so that the entire muscularis will contract synchronously. Then the distension and gastrin increase will act on the distension, and also the hormone gastrin will increase the force of contraction. Contractions are very vigorous and powerful, quite close to the pylorus end. And 30 ml of chyme is actually produced, okay, either delivered like in 3 ml spurts into the duodenum, and the rest of the 27 ml forced back into the stomach. So only liquids and small particles are allowed through the pyloric valve. All right, so right here is pretty much part of the humoral activity that is discussing sort of like a mechanism of acute allergic and hypersensitive response, allergic response. Uh, hmm, I think this is just, uh, I think this is just, let's try to eliminate this from our discussion. I don't think this is included. I do not know. This is probably a, what do you call this? An accidental addition which is not supposed to be part of our discussion. So let's take a look at the peristaltic waves, or the movement of muscles in the stomach to empty it. Okay, first of all, you have propulsion, which is the peristaltic waves that move food from the fundus towards the pylorus. Then you have the grinding effect, which is actually the more vigorous, as a matter of fact, most vigorous peristalsis and mixing action that occurs quite close to the pylorus. The pyloric end of the stomach here acts as a pump that delivers small amount of chyme into the duodenum. Okay, and then you have what you call the retropropulsion. The peristaltic wave here closes the pyloric valve, forcing most of the contents of the pylorus backwards into the stomach. So what regulates the motility of the gastric emptying? Well, pretty much your duodenum can prevent overfilling by controlling how much chyme can enter it. There are duodenal receptors that respond to stretch and chemical signals. And then you have also the enterogastric reflex, the enterogastrone that inhibits gastric secretion and duodenal filling. The stomach empties from a full meal in about 4 hours. But if you have an increase in fatty acid that enters the duodenum, that can increase the time of stomach emptying into 6 hours or more. Usually, carbohydrate-rich chyme will move quickly through the duodenum, more than your fatty-rich chyme. Okay, so pretty much that's actually something that we have already discussed. Let's take a look at the homeostatic mechanism. A few things: clinical homeostatic imbalances in the stomach. Vomiting is caused by extreme stretching of the stomach muscle wall, and also caused by intestinal irritants such as bacterial toxin, excessive alcohol, spicy food, and some drugs. Okay, and pretty much here it is caused by chemical and sensory impulses that stimulate the emetic center of the medulla. And excessive vomiting can actually lead to dehydration and electrolyte imbalances, which you call alkalosis. So now we go into the accessory organs inferior to the stomach. You got your liver, your gallbladder, and your pancreas. These are accessory organs that are associated with the small intestine. First of all, the liver, which is the biggest organ in the abdominal cavity, but only second only to the skin. The skin is the largest organ of the entire body. But the liver is the largest in the abdominal cavity. It is responsible with its digestive function in the production of bile. Bile is an emulsifier. Bile is pretty much like serving to emulsify fat loaded chyme before we digest it. The gallbladder is the chief storage organ for bile, and then the pancreas supplies most of the enzymes that's needed to digest chyme, as well as bicarbonates to neutralize the stomach acid. So here's your liver. The liver is made up of four lobes. You got the right lobe, the left lobe, the caudate lobe. This is the end. And then you have the quadrate lobe. It's the largest gland and it weighs about 3 pounds. Okay, it consists primarily of the right, left, caudate, and quadrate lobe, which I already uh, mentioned. The falciform ligament, and that's your falciform ligament right there. Okay, separates the right and left lobe, suspends the liver from the diaphragm and the anterior abdominal wall. The round ligament, this is actually the round ligament. The round ligament or ligamentum teres is a remnant of the umbilical vein along the free edge of the falciform ligament. So the falciform ligament is a ligament that goes all the way to the top. Okay. So lesser omentum. This is again your lesser omentum, the same picture that I showed you earlier. The lesser omentum, that green area right there, anchors the liver to the stomach. The hepatic artery and hepatic vein enters the liver at the porta hepatic. These are all, I'm afraid these are all lab, and therefore I'm just leaving it to you to be identifying, okay, in the lab. And take a look at the cross section of the liver. On a histology, on the point of view of histological point of view, your livers are made of hexagonal lobules. Okay, and these hexagonal lobules pretty much are would have specialized portal triad on each corner. Okay, they are made up of plates of hepatocytes that filter and process nutrient-rich blood. So that's it's another picture of that. And here is an up close picture showing you the central vein right there. Okay, and the connective tissue septum that is supposed to be making it visible in a good slide of the liver. You will see really that hexagonal lobule. So the portal triad in each corner, okay, in each corner of this particular hexagon-shaped lobule, you will see a branch of the hepatic artery which supplies oxygen, a branch of the hepatic portal vein which bridges nutrient-rich blood from the intestine, and then a branch which will carry bile from the bile canaliculi, and you call that the bile duct. I don't have anything here that shows the portal triad, but it shows us the central vein. But you could see here in this picture here, it's a three-dimensional diagram of the portal triad, rather lobule, and you could see that this is a triad right here, a portal triad. It'll be made up of an artery, a vein, and the bile. So every corner would have it. You got an artery, a vein, and a bile duct, and artery and a vein, a bile duct. So there's always a trio right in the corner of each. This is an entire lobule, and right in the middle of the lobule you will have the central vein. Okay, just know that you will have to know this in your lab, in your lab practical, which is probably what you're doing today. So pretty much in the microscopic anatomy, let's know that the hepatocytes have increased number of rough endoplasmic and smooth endoplasmic reticulum, Golgi apparatus, peroxisome, and your mitochondria because of the nature of the function of the hepatocyte. And pretty much it produces 900 ml of bile per day. It processes uh, blood-borne nutrients such as glucose being converted to glycogen and then making plasma protein. The liver is a very metabolic organ. It stores fat soluble vitamins, it performs detoxification. Okay, an example of this will be converting ammonia to urea. These are some examples only of what liver can do, and we are going to be discussing it in the next chapter in nutrition. So the bile, okay, is yellow-green alkaline solution containing bile salts, bilirubin, and pretty much cholesterol, triglyceride, phospholipids, and some electrolytes. Bile is pretty much like part of what you call goes around the enterohepatic circulation. It is recycled by a recycling mechanism that aims to conserve bile salts. And the bile salts are reabsorbed into the blood by the ileum, which is the last portion or the most distal portion of the small intestine. And then it is returned to the liver via the hepatic portal blood. And then it is resecreted again by the liver and restored in the gallbladder. About 90% of the secreted bile salts are recycled, and only 5% is newly synthesized each time. And so this is your enterohepatic circulation, guys, and I'm going to leave that to you so that I will not be redundant. So there are some homeostatic imbalances of the liver. One of them is hepatitis, which is usually a viral infection that causes, or actually it's caused by drug toxicity, or it could be wild mushroom poisoning. And cirrhosis, which is inflammation of chronic inflammation of the liver. This is actually progressive, more chronic than anything, brought about by chronic hepatitis or maybe alcoholism. Okay, here the liver becomes fatty and fibrous, and pretty much whenever the liver goes, transplants are really successful. But then availability of liver is really very scarce. Liver can regenerate to its full size in 6 to 12 months after 80% of it has been removed. Gallbladder is a thin-walled muscular sac on the ventral surface of the liver. It is a storage organ that stores bile by absorbing water and ions. It contains many honeycomb folds that allow it to expand as it fills. Again, a muscular contraction releases bile via the cystic duct that flows into the bile duct. And here is actually some pictures of gallstones, and you could see how nasty it looks. It's caused by too much cholesterol or too few bile salts. And this can obstruct the flow of bile from the gallbladder. Boy, does it cause really lots of pain in the back. Okay. Okay. The pancreas. The pancreas is another very important, matter of fact, one of the most important digestive organ accessory. And it is retroperitoneal. It's deep into the greater curvature of the stomach, and the head is encircled by the duodenum, okay, and the tail abuts the spleen. The function of the pancreas that relates to the stomach is exocrine. It releases the pancreatic juice, and it has acini, or this group of cells, a cluster of cells that produce zymogen granules containing proenzyme, and then it's released into the ducts. Here you have a small duct, and into the duodenum via the main pancreatic duct. This is actually a histological picture of the acini of the pancreas that releases pancreatic juice. So the pancreatic juice pretty much is about roughly about 1,200 to 1,500 ml per day. It's very watery and alkaline. It has a pH of 8, the purpose of which is to neutralize the acidic chyme coming from the stomach. It has electrolytes, bicarbonates, and digestive enzymes, proteases, okay, for digestion of proteins, amylase, lipases, nucleases, okay, and pretty much proteases are secreted in an inactive form. They need to be activated when they reach the duodenum. Okay, and we are going to get into that when we get into the chemical digestion and chemical conversion of the things that we have in the digestive system. But right here, what is shown right here is that you have uh, the activation of inactive proteases by enteropeptidases. Okay, first of all, when it gets to be released by the pancreas, the inactive form of the protease is called trypsinogen and chymotrypsinogen and procarboxypeptidase. The trypsinogen is going to be stimulated to be formed to be activated into trypsin by membrane-bound enteropeptidases. The chymotrypsinogen is going to be converted into chymotrypsin by trypsin itself. And then trypsin also is going to be activating the procarboxypeptidase into carboxypeptidases. Once trypsin is activated, there's no telling. It's going to be like a chain of reaction. All of the proteases are going to be turned on. So the bile duct and the pancreatic duct is actually uniting in a wall of the duodenum. They fuse together in a bulb-like structure which is called the sphincter, called the hepatopancreatic ampulla, which leads into an opening or otherwise known as the sphincter of Oddi. Okay? And pretty much it opens into the duodenum, and that's where a pancreatic juice is going to be released into the duodenum. So right there, okay, this is your bile duct, and it connects with the pancreatic duct, and right here it forms the sphincter of Oddi, and it pretty much will be opening into the duodenum. Okay, so what regulates the secretion of bile and pancreatic secretion? Well, they are both stimulated by neural as well as hormonal secretions. Hormonal control would include cholecystokinin, secretin, okay, and bile secretion is actually increased when pretty much the enterohepatic circulation returns large amounts of bile salts. Secretin then, at this point, from the intestinal cells, exposed to HCl and fatty chyme, would now stimulate the gallbladder to release bile. And then the hepatopancreatic sphincter is closed, okay, unless digestion is still active. Bile is stored in the gallbladder and is released into the small intestine only by way of contraction. All right, and here you could see the CCK and secretin are secreted by the duodenal enteroendocrine cells, and both of them are right here. Cholecystokinin release is stimulated by proteins and fats in a chyme. Secretin, on the other hand, is released. Secretin release is stimulated by the acidic chyme, and cholecystokinin and secretin together will enter the circulation and cause the four events. Okay, pretty much the four events cause four events, which is actually the stimulation of the pancreatic secretion. Okay, CCK induces secretion by the acinar cells of the enzyme rich pancreatic juice. Secretin causes secretion by duct cells of bicarbonate rich pancreatic juice. And the vagus nerve uh, weakly stimulates during the cephalic and gastric phases. Bile secretion by the liver, as bile salts are returned from the enterohepatic circulation, it becomes a very powerful stimulus for bile secretion. This secretin here is a minor stimulus in the secretion of bile. Gallbladder, when gallbladder contracts, the gallbladder contraction pretty much is actually caused by CCK. Here, the vagus nerve stimulates the weak gallbladder contraction during the cephalic as well as gastric phases. Then finally, this particular hepatopancreatic sphincter would be relaxed. The CCK causes the hepatopancreatic sphincter to relax. Bile and pancreatic juice now will enter the duodenum, which is the final phase. And that's it for today, folks. And I bid you well. Have a good weekend.